A lecture. So as you know that in last lecture I talked about some concepts of the databases. In that we talked about some brief about the GenBank, ENA and DDBJ. So today we'll start, and I already explained the concept of the INSDC International Nucleotide Sequence Database Collaboration. So I told you there is three major nucleotide databases are there: GenBank, ENA and DDBJ. So if you suppose sequencing, right, along with the annotation, if you want to submit your sequence to the GenBank, then how you can? Definitely the GenBank is the primary database, and if you want to if you want to see the database, if you want to retrieve any information from the GenBank database, there is no separate website for the GenBank. You have to go over the NCBI. In the NCBI itself you will get the GenBank. And there are some practical videos I also shared with you. Hopefully now you are skilled with that how to do the query in the from the database, and that answer you should got, right? So hopefully I think each of you have watched the video. Any researcher want to know about any particular sequence which is available in the databases, how you can retrieve, right? So GenBank, if you want to see if you want to retrieve the information from the GenBank, then you have to go on the NCBI, the National Center for Biotechnology Information, and I told you that NCBI is the example of the composite database, right? So whatever the box was there in the NCBI in which you did the query, that box here the GenBank people or NCBI people do the naming, they nomenclate them, and that the box in which you put your query, that is known as entrej. So with the help of the entrej, you can retrieve any information from the GenBank. And if you want to submit any kind of information, any kind of the biological sequence to the GenBank, then how you can submit? For that you need some special kind of the programs, and that is inbuilt with the GenBank or the NCBI, and the name of that is known as Bankit. Just like, let me tell you, say for example, if you want to create your if you want to post something on the Facebook, then obviously until and unless you don't have the user ID and password of the Facebook, you will not be able to post anything on the Facebook. So for that one, what you have to do, you have to first of all you have to go on the Facebook, and you have to click on the sign up, right? In the sign up, it will ask you certain questions like what is your name, what is your phone number, what is your mobile number, whatever, what is your age, date of birth, right? So basic details Facebook will ask you, and once you will give all the information, it will Facebook will ask your preferred user ID and the password. So once your user ID and password is with you, now with the help of that you will be able to log in, and now you will be able to post anything on the Facebook, right? Same way, if you want to post any information in the GenBank, you should fill some basic forms, and once the user ID password is with you, with the help of one tool that is known as Bankit, with the help of the Bankit, you will be able to submit your sequence to the GenBank, right? So two things you have to remember. The first of all, what is the name of the retrieval tool through which you can do the query from the GenBank? The name of that is entrej. And what is if you want to submit any kind of the sequence to the database, what is the submission tool? Or if you want to update something in the existing entry, the submission tool is known as Bankit. Meanwhile, likewise, if you the ENA, the European Nucleotide Archive, if you want to see the ENA, then definitely you have to go for the secondary, composite database, that is EBI, European Bioinformatics Institute. If you want to retrieve something from the ENA, then again you need another entrej type of software, means the retrieval tool, and that is known as SRS, Sequence Retrieval System. And if you want to submit your sequence, just like I explained in the case of the GenBank, you need a separate program, and that is known as Webin. So please remember, the name of the retrieval tool in the case of the ENA is SRS, and the name of the submission tool for the ENA is Webin. Likewise, if you want to see the DDBJ, the best part is DDBJ is maintained by NIG, National Institute of Genetics, and there is a direct website of the DDBJ, ddbj.nig.ac.jp. and if you want to retrieve something from the DDBJ, then one retrieval tool is there, that is known as getEntry. And if you want to submit any particular sequence to the database, the name of the submission tool for the DDBJ is known as Sakura. So whatever the retrieval tools and submission tools I told you, that is very important for any kind of the competitive examinations in which bioinformatics have been asked, right? So you please do remember the name of the submission tool and the name of the retrieval tool for each and every databases. So as we already discussed about the some GenBank, the basics of the GenBank and the DDBJ and ENA. So now I'm going to tell you some more fact about the GenBank data. So full release every 2 months. What does that mean? Means say for example, if currently you see the number of sequences is deposited into GenBank, say for example it is 25 crore right now. So, and in this 2 month, in coming 2 month, whatever sequence will be deposited by submitter, that will not be reflected to the database immediately until and unless it is verified by the GenBank administrator. So once it is verified, then after 2 months that will be released. Then again 2 months there will be the curation will be going on in the background. Once all the sequences is verified, they also will be released, right? So after every 2 months, the GenBank is released. Incremental and cumulative update daily. Daily, whatever new sequences is being updated, that is no that reflected in the GenBank and available only through internet. If you don't have internet connection, definitely you will not be able to means use the services of GenBank or any of the database. Yeah, sorry for the interruption. So I was telling you that on every 2 months there is the full release of the database so that whatever sequences have been deposited in that particular time period that will be available to the public for the access, right? Okay. So the in a previous video, the whatever I shared for the practical point of view, in that one, no, I told you that some keywords was demonstrated, or there is two type of query you can make in the database. The first one was the SSN based, and second was the query based, right? So in the SSN number, if you do have, if you got the SSN number from any of the research paper, if you put that SSN number, then you can get your specific sequence from the database. And if you don't have the SSN number, with the help of the keyword based searching, you can search any of the protein sequence or gene sequence or anything, right? So whatever things, if you remember, to see to understand this particular concept, practical performance is very important. If you have not visited that particular video, if you have not watched that particular video and you did not follow those steps, believe me, this step, whatever I'm going to explain, you will not be able to understand, right? So the entire GenBank record, which you have means you watched in my video, The GenBank record can be divided into three parts. One is the header, feature, and the sequence. Header, feature, and sequence means what? In the header, whatever information will be there, that will be applicable throughout the whole record, right? And feature, whatever annotation of the sequences are there, that will be available on the feature, and finally the sequences. So let me show you one sequence. Say for example, or else what I can do, I can live demonstrate to you. I can live demonstrate to you so that you will understand more. Say for example, I am opening the website, the NCBI, the composite database, ncbi.nlm.nih.gov. Although it is going to take time, depends over the internet speed, right? So this is the NCBI, right? Here I don't have to do anything. Say for example, I have the keyword. I have the SSN number. So one SSN number I got from any particular research paper. So let me search the entry with that SSN number. So suppose I am putting here HQ190916, right? So definitely I told you that since SSN number is unique, so it doesn't matter in the database millions of sequences are there or billions of sequences are there, right? You will get only one sequence. So if I click on the search, see here, if I click on the search, doesn't matter how many databases are there in the NCBI. Since SSN number is unique, so I will get one and only one sequence. See here, this is the entry I got. See here, Salenococcus rogeus having the SSN number HQ190916. If I click over here, the detailed information regarding this particular sequence will be available, will be visible to me, right? See here, the Salenococcus rogeus strain RVS-COKH116S ribosomal RNA gene partial sequence. So, see here, the something is written there, right? Something is written there. So the entire record, starting from locus up to this double slash, can be divided in three parts. Number one, I told you the header, means starting from the locus up to the journal, right? Up to this one. This is known as what? Header. Means whatever things have been highlighted here, that is applicable over throughout the record. The feature means whatever annotation, the validation of that particular sequence is available, that will be available under the feature. And finally, from the origin, here directly you will get the ATGC. So that's how this is the bifurcation of the page, the header, the feature, and the sequence itself. Alright. So let's understand one by one. What does it mean? What does it mean by locus? What does it mean by definition, SSN, version, keyword? One by one. So in the locus, what is there? So in the locus, first of all, what happened actually? Once there is the starting of the GenBank, then people started submitting their sequence in the GenBank, right? So what researcher thought that day by day the number of sequences is going to be increased. So why not to fix such criteria so that each and every sequence will be unique, right? So that's why they have put at the locus. The heading they have put at the locus, because just like, let me tell you one example. Suppose someone name in the class is Abhishek, right? So if Abhishek, maybe class is big, maybe 200 students are there in a class, and name of the student is named as Abhishek. So once I call it as Abhishek only, maybe 2 to 3 students say that their name is Abhishek. Then if we say, okay, no, I need Abhishek Kumar, maybe number of students will be less. If say Abhishek Kumar Singh, the number is going to be further reduced. If I say, okay, if Abhishek Kumar Singh having date of birth this, then there will be only one student. So likewise, this locus is the combination of different things so that each and every sequence will be unique, right? So previously, in the starting of the GenBank, there was no concept of the SSN number. SSN number term came later on, just like we people know. Now we have started using last 3 to 4 years. Now we have started using the Aadhaar card number. Previously what happened? You have several identity cards, like you have the PAN card, you have the election commission, the given identity card given by the election commission, the driving license. Now everything is merged under the Aadhaar card, right? So, so now later after the starting of the Gen Bank, the SSN number concept was not given, but later on the SSN number concept came, right? So now whatever sequence you can see here, you can see the SSN number, but if you see the older sequences, maybe you will not get the SSN number here. Then number of bases, that whatever sequence have been submitted, how many number of bases are there, whether the submitted sequence is DNA, RNA or protein, whether it is DNA or RNA. If it is RNA, then mRNA or tRNA, or which type of RNA is there. So here it is the molecule type, here it is here is the length, sequence length, the molecule type, means whether it is DNA or RNA or whatever. Then the topology of the molecule, whether it is linear or circular. Then here it is this is the fourth one, is showing the division. What does it mean division? Let me tell you. So for example, in your college separate departments are there, right? So if someone asked from outside, and he is asking the question about he want to know much more about the viruses, the common person. So he will visit to the principal office, that I am the person, I am eager to know about much about the viruses. So principal, what principal will do? They will principal will instruct that person that to go to the microbiology department, and their professors are there. They will be they're going to help you, right? So likewise, once you have the sequence, I told you that whatever sequence you means submit in the database, without verification, they are not going to be reflected in the database. They are not going to be publicly available until and unless they're verified, right? So, and it's also not possible for the database administrator to visit each and every lab, that whatever sequence have been submitted, they are genuine or not. So what happened actually? So for the ease of the verification, what GenBank did, they made the 18 division, just like 18 departments in the university. So 18 divisions are there, right? So some division is known as BCT, means bacterial division. So if I submit any sequence which is from the bacteria, so my sequence will be going to the BCT division. So their scientists are there, they will take care of my sequence so that it is genuine or it is falsified. If if I have done the sequencing related with the viruses, then different division is there related with the virus, VRL, viral division. If I have done the sequencing of the plant and I have submitted, there is another division that is known as PLN, right? If someone work on the fungi, so there is the FUN, means fungi division. So the scientists working with the fungi division, they have means spent their life to work with the fungi only. So they are well skilled, they are well knowledgeable. They will come to know that whatever sequence you have submitted, that is genuine or not. They will verify it, right? So that's why there is total 18 divisions are there in the GenBank. So for the different entries, this information is going to be vary. This molecule length is going to be vary. The molecule type will vary, but all the information will be in the same order. On which date? What was the last? Means on which particular date the sequence was submitted? 6 September 2010. Then definition. In short, what is the definition of the sequence? You can see. So for each and every sequence, there will be different definition. SSN number, I told you, the unique identifier for any sequence. The version dot one. There is no difference between the SSN and version, except here in the version it is written dot one. What does it mean? So for example, if today I write a book and publish it, then automatically there will be the first edition, right? There is no need to say anything. That is by default, it is first edition. After 2 years, once I will release the new book with the same name, with the same content, with modification, then that is going to be called a second edition of the book. Likewise, suppose say for example, today I have submitted one sequence, right? And after the years of the experience and years of the experiment, I came to know that there should be some modification, means whatever I have submitted previously, there is the needs of modification. So I can request to the database administrator by giving the SSN number that I am the author of this particular sequence and I need this kind of the modifications to be changed in the database. So that's why it is known as a version, means by seeing the version you come to know that how many times there was change in the entry. Keyword. Keyword, as you know, by with the keyword, if you put the keyword, you can search anything from the database, and if it's optional. If the researcher have provided it, it will be available. If it is not, then this line will be blank. Source, from which organism the sequence was retrieved, the sequenced, from which organism the sequence was sequenced, isolated and sequenced. Then what is the, under the organism we will find the scientific name and the classification of that particular organism. Then here is the references. Under the references, how many research paper have been published based on that particular sequence, right? So here it is a reference. If suppose a researcher have published 5 paper, so reference, author, title, journal. Suppose if 5 papers are there now, so this reference to author, to title, to journal will repeat 5 times. And if there is no paper published yet, then only will be one reference. Say here you can see the reference, the author, these are the name of the author, title means direct submission, and the researcher belongs to Sri M and Rani Science College, Rajkot, Yogi Dham, Kalawad Road, Rajkot, Gujarat, something, right? So there is no kind of the publications related information is available there. If it was there, then might have in this particular title in the journal, the name of the journal would be there, right? So here researchers have directly submitted the sequences. They have not published the paper yet. Feature means, under the feature, whatever annotation is there, no, that will be available with the feature. Like what is the organism, molecule type, strain, country, PCR primer which they used for the amplification, then what is the ultimate product of that 16S rDNA. So this is the rRNA, and then finally the origin, and here you can see the direct sequences, and you can see here that there is total of 6 column, 1 2 3 4 5 6, and in each column there is 10 bases, right? So in each row there is total 60 bases. So likewise you can see that around 1441 means around how many bases are there? 1468 bases are there, and this one is always ends with the double slash. So this was the GenBank file format. This is known as GenBank file format. So in my PPT, I have explained each and everything. Excuse me. Excuse me. So in my PPT, I have explained each and everything in minor detail. So please go through it, and you will come to know about each and everything. Here one thing is very important. By if you read the GenBank file format, because I told you, in the practical session I demonstrated only the GenBank. I did not demonstrate the DDBJ. So if you are skilled with one database, definitely you will be able to do the second. You will be able to retrieve the information from the second database also. See here, let me demonstrate it for you. See here, if I want to see, because now there is no meaning of attending all these 3 databases. If you are skilled to how to retrieve the information from the GenBank, definitely the same information is going to be available with the ENA and the DDBJ. But for sake of uh, for sake of explanation, I am telling you how to retrieve the information from DDBJ. So ddbj.nig.ac.jp. See here. I'll share all this PPT to you. Don't worry. So whatever explanation I am doing, that I will make it available to you so that you can prepare. And the number of students who are joining the class, they are very less. I will talk to Anil sir. Why students are not sincere? Only 5 to 6 people are joining the class. Although the number of students, I was being informed that there is total more than 40 students are there, but only 6 to 8 students are there. I don't know how you will be able to write in the examination, right? So if you go on the DDBJ, say here the simple interface of the DDBJ, right? If you want to retrieve something from the DDBJ, how you can do? So here the first link you see, services, search analysis database service of DDBJ center. If I click on the service, see what I'm going to get. See, The database, no, the interface you may change. So it changes time to time. So as I already told you, if you want to retrieve any information from the DDBJ, one retrieval tool you will require, and that is known as get entry. So you try to locate where is the get entry. So here is the get entry you find. So if I click on the get entry, see what is going to happen. See. So now get entry is there. So if I click on, if I put the same SSN number which I put it in the GenBank HQ190916, just simply click on search and just wait for a second. What is going to happen? It will ask you about the English or Japanese. Something is wrong. Let me do it again. Data retrieval by SSN number. Let me check it out. Why it is not giving the answer? Yeah. Some. Let me do the query again. It is HQ190916. Yeah, see, HQ, once I put it HQ190916, directly whatever I got in the case of the NCBI, the GenBank, the same information I got in the form of in the database in the DDBJ database. So in your syllabus, if I show you your syllabus, in your syllabus, if you see here, let me clarify your doubts so that you will not get any confusion while you are seeing the syllabus of yours. So let me show your syllabus. In your syllabus, it is written here, Introduction to Bioinformatics, Application and Research in Bioinformatics, Present Bioinformatics Scenario, Characteristics of the Bioinformatics Database, Categories of the Bioinformatics Database means types of data, Sequence Database means nucleotide EMBL. Now the name of the EMBL have been changed, it is what? It is ENA. So very soon I am going to explain about the ENA also. The protein, here it is written protein, but that is wrong. Actually this is the nucleotide database only, DDBJ. So whatever the file format of there, for the GenBank, the same will be of DDBJ. So this is only just copy paste. So whatever the GenBank file format, the same ditto is copy pasted by DDBJ. So if in the examination it is asked that what is GenBank, then you have to write down the background of the GenBank, whatever I told you. What is the SSN number, means what is the retrieval tool, what is the submission tool, and file format regarding the file format. If it is asking about the GenBank, definitely that is not in your syllabus, so nobody is going to ask about the GenBank. So if it is asking about the DDBJ, so you have to write down the background information about the DDBJ, and whatever file format was there, for the GenBank, the same thing you have to write in the case of the DDBJ. There is no difference. So that's how we are able to finish today's DDBJ and DDBJ file format. So since the number of students are very less, so I'm not going to continue the lecture now. So we'll see you tomorrow at the same time, and then I will finish it up the ENA, European Nucleotide Archive.